Hello, and welcome to Let's Meet the Virologists, a podcast about the people behind today's virology headlines. People just like you working to understand viruses and how they affect you. During the COVID-19 pandemic, we are talking with postdoctoral fellows involved in coronavirus and COVID-19 related research so that you can learn who they are and what they do. I am Larissa Thackeray, and I am hosting this podcast from America's Heartland in St. Louis, Missouri. With SARS-CoV-2 cases and deaths continuing to rise, we are waiting to hear about the efficacy of monoclonal antibodies and vaccines for preventing or ameliorating COVID-19 disease. On October 21st, 2020, we talked with Dr. David Martinez, a postdoctoral researcher in the Barrick Lab at the University of North Carolina at Chapel Hill, who has been working to characterize novel therapies and vaccines against SARS-CoV-2. David obtained his PhD in molecular genetics and microbiology from Duke University, studying HIV, and has been working to understand how viruses such as flaviviruses and coronaviruses evade host immune responses. Hi, David. Happy to have you with us today. Why don't you tell us a little bit about yourself?
Well, thank you very much, Larissa. So I'm a postdoctoral fellow in Dr. Rolf Barrick's lab at UNC. I study viruses. I received my PhD from Duke University in Dr. Sally Permar's lab, where I studied immunity to HIV. And I moved over just across the street to UNC, and I began my postdoc in Dr. Barrick's lab.
How did you become interested in virology research way back when?
So I was sort of always interested in microbiology in general. So as a kid, my dad had bought me a microscope when I was probably about 5 or 6 years old. He was a physician, actually. And on the second story of his clinic, he actually had this diagnostic laboratory for just standard clinical diagnoses. And I used to play up there. So I was always sort of familiar with these machines that I had no idea what they did, but nevertheless, the lab was a place that I played. So he had bought me this microscope, and I was always just sort of interested in nature and microorganisms in general. So I guess that's my earliest memory of me being interested in microbiology. And senior year of high school, I had decided that I was going to attend the University of Oklahoma, and they in fact had a microbiology degree program. So I was a micro major from day one, and I became really interested in viruses, in particular my senior year of undergraduate, where I took this virology course with a couple of virologists, in fact, at the University of Oklahoma. And I just became fascinated how these tiny little, essentially like tiny little genomes could really just manipulate the cell to do these crazy things. And I just wanted to learn more about them.
Cool. And can you tell us a little bit more about sort of the steps that you took from undergraduate to graduate and so on to sort of get to where you are in your career today?
Yeah, so I guess began my first formal maybe training in virology in Dr. Robert Kaleda's lab at the University of Wisconsin-Madison during an undergraduate summer research program. And there I was studying cytomegalovirus proteins and how they modulate immunity and sort of infectivity in human cells. So that was really my first sort of experience with what I would call real virology in a lab, and really doing more cellular biology in sort of the context of viruses. So I think after that, I sort of became interested in maybe back then was, and even until I would say 2020, what in my mind I thought was sort of the holy grail, or the coolest virus, because it was the most difficult one, and that was HIV. So I researched which programs had really just really active in top HIV research, and really just based on that, I selected which schools I would apply to, and I ended up at Duke. Actually, they had for a number of years, some really active work in this sort of the HIV vaccine side of the HIV field, which is now, I mean, until recently, it's actually now what's sort of maybe still on the up. I mean, sort of the pathogenesis didn't really exist anymore. There's only a couple of labs that were doing that.
Right.
So yeah, I mean, it was sort of I wanted to study maybe the hardest virus to that in my mind I thought was the hardest virus, because here we were 30 years later with still no vaccine, and we still don't have a vaccine. So that's how I came to Duke University to study HIV.
And how about when you were looking for your postdoc? So what was the next step for you?
Yeah, so I was still interested in viruses, and I knew that I didn't want to do HIV anymore. So that was a really good training, I think, for me. But I was sort of ready to shift gears, and it's funny because I thought the HIV field is almost saturated and hyper competitive. I almost thought like you have to be part of a handful of groups, and you sort of have to have a senior role in these groups to be able to do I think really meaningful studies. No offense to everybody else, but this is sort of where the conclusion I came to at the end of my PhD. So I thought, well, I need to pick something that's still important, and maybe not as saturated and competitive at the very high levels. So I came to the Barrick lab to study flaviviruses, and I still study flaviviruses. In fact, just had my first postdoc paper published on flaviviruses, that is, earlier this month. And, funny enough, right, I left this hyper competitive field, and being in the Barrick lab, of course, we've all been happily, I think, sucked into the COVID-19 work. So I left probably what I thought was a highly competitive field to perhaps the most competitive field that I think I should be careful in saying this, but I would be very surprised if it's not the most competitive field out there, at least for biomedical sciences, at least at the moment. As of October 21st of 2020, we talk to the editors, right, of whenever we're getting ready to submit a paper, and they tell you we're interested today, but this could change by the time we get the reviews back, kind of thing. So that's how I picked, the Barrick lab was, I still wanted to stay in viruses, but not necessarily HIV. And now I'm sort of at this emerging virus interface, both of studying both flaviviruses and coronaviruses.
Right. So to follow up a little bit more on that, can you tell us about this work that you just published on dengue, I think it was, right?
Correct. Yeah, so that was a project that I initiated early on whenever I began my postdoc here in the lab, and that was at least for me. Of course, maybe everybody thinks that about their paper, but what I think that paper contributes to the field, and sort of what maybe it's like, I'll just give you like the 30,000-foot view. Is that historically in the dengue field, there is sort of this dogma or sort of like traditional thinking that immunity to a single serotype is generally broadly protective against that serotype. I wouldn't say essentially forever, but for a long time. And what this study really showed was that there's a great diversity, genetic diversity, within a single serotype. In fact, if you were to break up the genotypic diversity within different genotypes, what we demonstrated was that this genetic diversity can really alter the ability of dengue virus to evade neutralizing antibodies in humans. So neutralizing antibodies, in fact, in the setting of vaccines, some dengue vaccines that are now in advanced clinical trials. So I think it really underlines that, we need to, I wouldn't say necessarily stop what we're doing, but sort of think about it a little more carefully in how we're designing the current dengue vaccines, because in my opinion, they're not really being designed with this question in mind, in how we can target all of the different genotypic variants within a serotype. So I think that's really what this study shows.
Cool, cool. And you mentioned that you obviously have been doing a lot of work with SARS and COVID-19 related research. Can you tell us a little bit about that work?
Yeah, so I would say, sort of for the first, maybe 6 months or so in the Barrack lab, I was at least, I'm sorry, let me rewind. Maybe within the 6 months or so since COVID-19 really took off in the world, really, because we started working on COVID-19 along with many other groups once the genome was released in China. So for the first few months, I was really sort of playing an ancillary role and really trying to help the clinical products get off the ground. So in fact, with a collaboration with you, Larissa, with the Crow Lab, then that worked really focused on isolating and characterizing antibodies that could be protective in animal models. And those antibodies have now been licensed to a major company, AstraZeneca. And through work with other collaborators that came through our lab, sort of, then we've been really involved with two of the Operation Warp Speed vaccines, both Johnson & Johnson, a lot of the non-human primate work, as well as the Moderna vaccine, both the mouse and the non-human primate models of infection. And really sort of, trying to help these major products move forward. Also the Eli Lilly antibody, we played a role in that as well. So the first few months was, really just trying to get my bearing in the BSL-3. I had actually just completed my BSL-3 training in March of this year. So, the timing was just absolutely perfect as far as, being able to, to get on these studies, but I would say really, just try to contribute to the public health effort. And I would say, the last few months, and I've sort of pivoted to my own COVID-19 work and pushing forward my projects. So hopefully in the coming months, then we can share a lot more details about those.
Great, great. And what's it like actually working on something that's so immediately relevant on public health? So in virology and immunology, often you work many hours for long periods of time, and the relevance of it is, years later, if you even get that. So what's that like?
Well, I would say, for me, and I've always sort of had this mentality of always in the back of my mind thinking, how does this benefit public health in general, and how do I set up experiments so we can try to have an impact on global health. So for me, it's been extremely rewarding just sort of seeing these products take off in the clinic, and really, at least the early data that we see in animals, looks really good. So for me, it's been an incredibly rewarding experience, and has really just re-cemented that initial desire to contribute to public health. So, I mean, I'm obviously probably like most people on your podcast, really nerdy and into science. But as much as I love science, I still want my science to be meaningful to the taxpayers that are actually funding the work that we do here. So it's been really just a rewarding experience. And I think maybe that's been underlined by, I think most virologists are in super high demand now, but by the constant stream of questions from family members and from friends, that I really think are just curious about what's going on. So for me, it's been really cool to see the work that we do have a major impact.
Cool, cool. And I guess sort of more generally, so up in your career so far, what has been the most exciting sort of Eureka science moment?
Yeah, so, I think you definitely have that all the time in the lab of, this tiny moment of discovery where you're the only person in the world that knows, understands a certain process. So, for me, this was, and actually my first PhD project that I did and published. And, that's still probably my favorite paper from graduate school. It certainly was not my highest impact paper, but it's still my favorite because I sort of remember, this long process that I went through and sort of worked really hard to get there, So for me, it was actually mapping, and that this is probably not exciting for a lot of people, but for me it was, and it still is. Whenever I read this paper, and it's sort of nostalgic, that I'm like, oh my gosh, like this is, this was like the first 3 years of my PhD or what have you. But it was mapping this protective antibody response to this region on an HIV glycoprotein, which is GP120, but the specific region was the variable loop 3, so the V3 region. So I had actually done extensive mapping, and once I received analyses back from the statisticians we were working with, then my PI and I had actually sat down. So maybe PI means the head of the laboratory for people out there that may not know what a PI is. So we sat down and we were reviewing this data, and once we were interpreting the results from the statistician, that it became clear that we had mapped it to this very specific region within the V3, and particularly this amino acid residue 317 to this phenylalanine, which at the time was really cool for me, to be able to map it down to a single amino acid residue, that this protective immune response, antibody response, in fact, in HIV infected women that did not vertically transmit the virus to their infant. So for me, it was cool. And I would say that was maybe, a really memorable sort of discovery moment in my scientific training.
What about, so conversely, what has been the most difficult thing you've had to overcome as a scientist so far, and how did you overcome it?
I would sort of just say, maybe it's just sort of the process of science. I think for me, maybe to my own disadvantage is that I get impatient sometimes, and sometimes the pace of my work, even when you think you're working really hard, but sometimes you're just not satisfied with the pace at which the science is actually moving. You wish you had the answers faster than you do. So for me, that's sort of been maybe what I would say is perhaps my biggest struggle. I don't know if I would pinpoint it to a certain time where things weren't working, but of course everybody has dry spells. But I would just say maybe just being impatient. But maybe I could pinpoint it to a time. I would say maybe after my preliminary exam defense and during my PhD, I was sort of really discouraged because I was, I'm really hard on myself, and I didn't think I did as well as I thought I should have done, and of course I passed and everything, and, everybody said it was fine, but I didn't think it went, perfectly like I wanted it to. So I was sort of really discouraged, and was even, sort of planning my exodus after my PhD to, no offense to anybody, because my wife is in industry, but that I was going to go to industry and just sort of be done with all these things. But then you get a cool finding in the lab, or you you have one good idea out there, and then you see a paper that is published a few months later, and it's something similar to what you were thinking, and then that sort of brings you back up, and you get excited again. So
yeah. So I guess sort of more globally, if you had a chance to ask your older self, say you when you're 60 or 70, one question, what would it be?
Well, I don't know. I feel like most people who think about this, or maybe who have an answer for this, I don't necessarily think they talk about work per se. I don't know. I would hope that, I would be able to ask myself if I was a good person, and made prioritizing things in life that are actually priority. And I think, maybe once you reach a certain age, like 70 or 80, then you sort of realize what the real priorities are. Hopefully you realize it earlier. But I don't know. I just really hope that that I'm able to have the same priorities then that I have now, and I hope that those align, because hopefully it means that I prioritize my time correctly on my short time on this earth.
I guess to follow up on that, so people often talk a lot about work-life balance, things like this. What do you do to achieve that? And how has that changed potentially during the COVID pandemic?
Yeah, so everybody obviously that works on COVID, they work pretty hard, I think, and long hours. And I wouldn't necessarily say that it's always a long thing, but it's more of a constant thing. It's always sort of this marathon, right? That you always have something on the weekend to do, even if it's a half day, but it's always, having to come in and always sort of be here. So for me, one thing that has really helped me sort of escape reality maybe is going on a daily walk with my spouse. So my wife and I will take a 30-minute walk around the neighborhood, and we just really try to talk, just talk through our days and just catch up on life in general. And it's a good time for me to just unplug and not really think about my experiments and the results that I got and sort of like what my next steps are. And along those lines, is on Sunday morning, we typically try to go on a hike to local place, pretty nearby areas here. So that's one mechanism for us to unplug from what's happening with COVID and try to reset your brain a little bit. So that's been it for me.
Yeah. So I guess to follow up on that, as a virologist, how do you make decisions about how to keep yourself or your family safe? So how do you make decisions about risk for various activities that you have to do?
Yeah, I would say it's probably, maybe no different than things that people can readily read on maybe like the CDC website, but maybe how it's a little bit different for us is that maybe we have access to that information before it becomes widely accepted, for example. So an example that I'll give is that early on in the pandemic, sort of before the whole mask thing became widespread, is we were looking at the infectivity of the SARS-2 coronavirus for the ability to infect cells in the nose. And once it became clear from the data that was coming off live from our lab that nasal cells were in fact more permissive to SARS-2 coronavirus, then I think that really lended support for widespread mask use. So I wouldn't say that maybe as a virologist I have maybe access to information that other people don't have, but maybe we have access to that information a little bit earlier because we sort of see what's happening in the news or what's happening in our lab really.
Do you find though that it's easier to sort of evaluate, so for example, like going out to restaurants or traveling or sending your kids back to school, do you think it's potentially easier to evaluate those risks being a virologist?
Yeah, absolutely. I think, not only a virologist, but I would just say someone with, that's science oriented. I think most scientists could maybe, at least biomedical scientists could maybe come to some of these conclusions. But I would say you're more hyper aware of things not to do. Like for example, I'm just maybe a little more of a germaphobe than I normally would be because of my virology training, maybe. And actually, yeah, maybe I could really see that because my wife is also a scientist and now in industry. But I would just say sometimes the way we disinfect things or maybe, whenever I've seen her handle stuff, I'm like, well, you should really do it like this, because if you were in the lab and you were removing these PPE, this is the way you'd want to do it to not contaminate or not try to do anything like this. So maybe that's like, one specific example, but I would just say maybe you're just a little more hyper aware of germs around you and what could potentially be contagion.
Yeah. What about, so with your family or your friends, do you find that people are reaching out to you with questions or, wanting advice on certain things?
Mm-hmm. Yeah, I would say all the time. I almost get, I feel like, daily questions from family members and or friends about developments in the news. And then conversely, right, not only do you get questions, but, sometimes you feel like, your family members, as well-intending as they are, they'll tell you something that, because of the work you're doing, you knew months ago, right? Or even years ago, because you're a virologist, right? Like, it's funny because, I've had a family member explain to me that the coronavirus causes this really inflammatory disease in the lung. To which I nicely agreed with him, and it's like, yeah, that's what it does. I didn't want to tell them, that we've actually known this for many, many years, because coronaviruses have in fact been around a long time, and this has sort of been known for a long time. But so you kind of see both, right? I mean, of course they're well intending, but you sort of see both sides of it. So you get really really good questions, about I feel like recently I've gotten a lot about the pause of both the Johnson & Johnson and the AstraZeneca vaccines, and why they're being paused, and you try to do your best, with the ability, with the information you have. So at least right now with the Johnson & Johnson, we don't know that the adverse event was potentially related to an individual that received the vaccine or not. I don't think we know that information, but that's sort of been those are the types of questions that I get a lot from family members, is just about where these products are in clinical trials, to the occasional explanation of something we've known for years. It's always comical, I just try to take it in a good light.
Yeah. So we're winding down. Do you have any last messages for our listeners? Any thoughts about the future of the COVID-19 pandemic?
Well, I would just maybe, to end on an optimistic note, I would just say that, hopefully the vaccines work as well in humans as they do in animal models. And hopefully we learn a little more about the durability of these vaccines. So that's sort of a big question out there, is, how long is this protective immunity going to last? And hopefully we can, as a field and as citizens and as people of the world, just collectively learn together. But, I would just say there are a lot of things in the pipeline that should hopefully help to control the COVID-19 pandemic, and hopefully things will turn up for the better in the coming months.
Great. Well, thanks so much. David has been hard at work over the past couple months evaluating new therapies and vaccines for SARS-CoV-2, and is hopeful that these treatments will work in humans based on the results he has seen from animal models. This has been Let's Meet the Virologist, a podcast about people who study viruses. This is your host, Larissa Thackeray, and thanks for listening. You can find us on Google, Apple, and Amazon Music podcasts, or at lmtv.podbean.com. If you are a virologist interested in sharing who you are and what you do, please contact us at letusmeetthevirologist@gmail.com.